Hello everyone, let's continue our discussion about antimicrobial treatment. So this is the second part for the video. So if we discuss antibiotics, those are drugs that are going to be effective against bacteria. Now we go ahead and we divide our antibiotics into two groups: broad spectrum antibiotics or narrow spectrum antibiotics. Broad spectrum antibiotics, those are known to be effective against more than one type of bacteria. So those broad spectrum antibiotics, they're going to be effective against gram-positive and gram-negative bacteria. An example of a broad spectrum antibiotic is tetracycline. Now, narrow spectrum, on the other hand, narrow spectrum, those are antibiotics that are going to be effective only against one group of bacteria, either gram-positive or gram-negative. Now, the thing is, though, some narrow spectrums, they are going to be, so for example, penicillin, okay? Penicillin is effective against gram-positive, but you will see that penicillin can still, be effective against a bit, some gram-negatives. Not a lot, and so therefore we don't consider penicillin to be a broad spectrum because, again, they are used to treat gram-positive infections and very few gram-negative. So that's why, again, it's not going to be considered broad spectrum. So that's one thing. Like if you guys see a narrow spectrum drug and you see that, wait a second, I can see that it can kill both gram-positive and gram-negative. So how come it's narrow spectrum? Because again, if it's effective mostly against, a major group, then we refer to it as a narrow spectrum. So penicillin, they're mostly used, against gram-positive bacteria, very few gram-negatives. So that's why it's a narrow spectrum. Polymyxin is a narrow spectrum antibiotic and it works against gram-negative bacteria. So we'll go ahead and discuss how these antibiotics work, and we're going to go ahead and take a look at a graph together. So let's go ahead and do that. If we take a look at this chart right here, you have a list of antibiotics. Let's go ahead and take a look at a few of them. So polymyxin is an antibiotic that's going to target the plasma membrane of the bacteria. And you can see that polymyxin is a narrow spectrum antibiotic. It targets mostly the gram-negative bacteria. For example, Pseudomonas aeruginosa, and they usually prescribe it when individuals have UTI infections. There is another one right here called carbapenems. Carbapenems is an antibiotic that's going to target the cell wall of the bacteria. So it blocks the cell wall synthesis or its repair. So at the end, it's going to be targeting the cell wall of the bacteria. And carbapenems, it's a broad spectrum antibiotic. It can target both gram-negative and gram-positive bacteria. Tetracycline is also another broad spectrum antibiotic, and tetracycline, it targets the protein synthesis of the bacteria. It inhibits the protein synthesis of the bacteria by actually targeting the ribosome, specifically the 30S subunit of the bacteria's ribosomes. If we take a look at penicillin, penicillin is a narrow spectrum antibiotic, and penicillins, they target mostly gram-positive bacteria, and specifically like gram-positive coccus bacteria. So, examples of gram-positive coccus bacteria are the ones that can cause strep throat and staph infections. so Streptococcus and Staphylococcus, genre. Now, penicillin can target a bit of gram-negative, but not a lot, and so that's why penicillin is still considered a narrow-spectrum antibiotic. But what we're gonna take a look at later is in the lab, they were able to take the natural penicillin and they were actually able to modify it to make it more of a broad spectrum antibiotic so that it can go ahead and target more gram-negative bacteria. But if we're talking about, the natural penicillin, then it is a narrow spectrum antibiotic targeting mostly gram-positive coccus bacteria. Let's take a look at what could be targeted by antibiotics. So there are antibiotics that target the cell wall. They either block the synthesis of the cell wall or they block its repair. And again, that's how the bacteria could be inhibited or even killed. Examples of some cell wall, inhibitors would be penicillin. Let's take a look at penicillin together. Penicillin, as we said, it's a natural antibiotic that's actually extracted from the fungus Penicillium. Penicillin, it's a narrow spectrum antibiotic targeting gram-positive cocci bacteria. But what they did in the lab is they took the natural penicillin and they were able to modify it in the lab so that it could target a bit more of the gram-negative bacteria. So let's take a look at the different versions of penicillin that they made. So this table right here kind of shows you a list of the penicillin antibiotics. So penicillin G and V, those were the natural, antibiotics that they've extracted, from the Penicillium fungus. So they tend to refer to them as the first generation. And you can take a look at the penicillin G antibiotic and how it looks like. And penicillin has this beta-lactam ring, which is like right here. You can see that beta-lactam ring. Now, penicillin G and V, those are, again, the natural versions of the penicillin. And as we said, penicillin is a narrow spectrum antibiotic targeting gram-positive bacteria. What's the difference between penicillin G and V? Penicillin V gets absorbed better, through the colon, so it has a better absorption rate. Now, are there some bacteria out there that are actually resistant to penicillin, meaning penicillin is not going to be effective against them? Yes, there are. So there are bacteria out there that are resistant to penicillin. And how can a bacteria be resistant to penicillin? If the bacteria, for example, carries an enzyme called penicillinase, and that enzyme, what it does is it breaks down this beta-lactam ring right there, making the penicillin ineffective. So definitely, if the bacteria has this enzyme right there, then you cannot use penicillin to go ahead and kill it. Are there some people out there that are allergic to penicillin? Yes. So if the individual out there is allergic to penicillin, then you can't give them like penicillin, so you have to think of another antibiotic to give them. These are some, other versions of penicillin out there: methicillin, ampicillin, amoxicillin. And so these are, some other versions of penicillin that they kind of modified and tweaked and tried to, tweak it in the lab. So for example, ampicillin, it's going to be a bit more of a, broad spectrum antibiotic because they went ahead and they, so ampicillin, before I say what it does, it's a semi-synthetic drug. Semi-synthetic because They took the natural penicillin and they tweaked it in the lab. And the reason why they did that, because as we know, penicillin targets mostly gram-positive bacteria. And so ampicillin can target gram-negative. So ampicillin is going to be able to target both gram-positive and gram-negative. And so it can also work on gram-negative bacilli bacteria. And so that's why we say that ampicillin is more of a broad spectrum antibiotic. But same, if you take a look at, ampicillin, well, we don't have a picture of it in this, table right here, but it's also going to have that beta-lactam ring. So again, one more time, if the bacteria has the enzyme penicillinase, then again, it's not going to be effective against that bacteria, because if the beta-lactam ring breaks down, again, then the penicillin, or in this, the ampicillin is not going to work. Amoxicillin is another one. It's, another version of the penicillin. It's more broad because it's also going to target now both gram-positive and gram-negative infections, and it has a pretty good absorption rate. Now there's actually another enzyme other than penicillinase. There's another one called beta-lactamase. So beta-lactamase is another enzyme that can also break down the beta-lactam ring. So if bacteria out there carry this enzyme, then again, all the penicillins out there, they're not going to be effective, because that beta-lactamase is also going to break down that beta-lactam ring that you see right there. So beta-lactamase and penicillinase, those are enzymes that break down the beta-lactam ring, making the bacteria resistant to this antibiotic. Other than penicillin that can target the cell wall inhibitors, we have the carbapenems. That's another one. And carbapenems, it's a broad spectrum antibiotic because it can target both gram-positive and gram-negative bacteria. Another one is vancomycin. Vancomycin, it's narrow spectrum. Okay, so vancomycin, it's a narrow spectrum antibiotic. It's usually used to treat staphylococcus infections. So staphylococcus, those are gram-positive bacteria. And since the vancomycin also targets gram-positive cocci bacteria, this could be an option to give to patients that are allergic to penicillin. So if someone is allergic to penicillin, which targets gram-positive cocci bacteria, then another option then the physician might think of is vancomycin, which also targets gram-positive, bacteria. If the patient is also allergic to vancomycin, then they will have to think of something else. Bacitracin is another one. Bacitracin is also a narrow spectrum antibiotic. Now, bacitracin is usually used to fight off skin infections. So skin infections, again, caused by Staphylococcus bacteria or Streptococcus bacteria. So again, that's why it's a narrow spectrum, because only targets these gram-positive, infections, Streptococcus infections, Staphylococcus infections. And by the way, this is the main ingredient that you find in the Neosporin antibiotic. So the Neosporin antibiotic that you apply on your, on your skin, that's the main ingredient right there. Now, there are, some that target the cytoplasm. So the these antibiotics, they target the cytoplasm, and that's how they can, affect the bacteria in a negative way. An example is polymyxin. And we mentioned polymyxin before, and polymyxin is a narrow spectrum antibiotic. It usually targets gram-negative bacteria. There are also antibiotics that can target basically the DNA of the bacteria. So these antibiotics, they can actually prevent the DNA from replicating, or they can go ahead and mess up with the process of transcription and translation, and therefore, again, they would go ahead and mess it up. By the way, other than antibiotics that can target the plasma membrane or the DNA replication, transcription, etc., are there other, antimicrobial drugs that can also target these things? Absolutely. Antifungal drugs. So antifungal drugs out there can also target, the cell wall of the fungus. They can also target the plasma membrane of the fungus. They can also target the DNA of the fungus. So other antimicrobial, drugs can also target these, same parts. But of course, you cannot use an antibiotic to treat a fungal infection, because there are differences between the cell wall of a bacteria and the cell wall of a fungus. There are differences in the plasma membrane of bacteria versus fungi. So you cannot use an antifungal drug to treat a bacterial infection. You still have to use, the correct medication, of course, because again, there are structural differences between a fungus and a bacteria. And I mean, if you think about it, fungi are eukaryotic microorganisms, so definitely there are a lot of differences between bacteria and fungi. But I just wanted to mention that also antifungal, medication out there can target, the plasma membrane or target the DNA, RNA, process, okay, of transcription, translation, DNA replications, etc. Now, another thing that could be targeted is the ribosome. Now, again, ribosomes are actually found in both bacteria and fungi, but bacteria ribosomes and fungi ribosomes, they are totally different. Again, remember, bacteria are prokaryotes, fungi are eukaryotes, so the ribosomes are different. Now, the ribosomes could actually be targeted, by antibiotics. So if we go back to the list of, antibiotics, that could target the ribosomes, we have, for example, let's take a look at tetracycline, since we were just talking about it in the previous chart. So we said that tetracycline, it's actually a broad spectrum antibiotic, and as you can see, tetracycline, the target is the ribosome, specifically the 30S subunit. And the tetracycline, basically by, kind of targeting that part of the ribosome, I mean, what is the big picture? It's going to, affect the protein synthesis. It's going to inhibit the protein synthesis production by acting on the 30S part of the ribosome, or the 30S subunit of the ribosome. And tetracycline is a broad spectrum antibiotic because it's going to be effective against both gram-positive and gram-negative bacteria. So tetracycline is, one of them. There is, let's take a look at another one. So there is azithromycin. Okay, so let's take a look at this one right here. Azithromycin, that one also inhibits the protein synthesis, but it targets another subunit of the ribosome. So azithromycin targets the 50S subunit of the ribosome. And azithromycin is also a broad spectrum antibiotic. So usually they use, so again, it can target both gram-positive and gram-negative. And azithromycin is, it is semi-synthetic, okay? And they usually use it to treat like ear infections, respiratory, skin infections. So azithromycin is another one. Now, what's another thing that could be targeted? The mechanism that happens like inside of a cell, so metabolic reactions, blocking certain pathways, by targeting certain enzymes, and so therefore the whole entire metabolic pathway comes to a halt. So again, that could be another thing that could be targeted within the cells, affecting, of course, the cell in a negative way, and therefore causing its inhibition or causing its death. So those are, things that get targeted by these antimicrobial drugs. The cell wall, the plasma membrane, the process of replicating the DNA or going through transcription translation, and then targeting the protein synthesis, and then lastly, targeting certain metabolic pathways within the cytoplasm of the cell, inhibiting it, etc., blocking the pathway. Let's take a look at this slide right here and kind of explain what's happening right there. So we know that we have our natural normal biota, or you can refer to it as your normal flora. And that means that, within us, inside of us, we do have bacteria, and they are part of our normal flora. So, for example, if you take a look at your colon, and, that's where we have, our normal flora. So you would find that in your colon, you have your normal biota, which consists mostly of harmless or beneficial bacteria. So some bacteria out there, they're not going to harm you. And then some bacteria that we have, they're actually pretty beneficial. They actually help us digest our food. They actually help us, clot the blood. So they're pretty helpful, and they're part of our normal flora. Now, actually, also part of your normal flora is to have some pathogenic, bacteria also in your colon. You're like, wait a second, pathogenic? So how come I am not sick if I do have some pathogenic, which are, again, disease-causing bacteria? The reason why you're not sick, even though you do have some pathogenic bacteria in you, it's because these normal, harmless or beneficial bacteria are basically keeping them in check. So they are preventing those pathogenic bacteria from taking over. They're preventing them from actually replicating in such high numbers, and therefore, as long as they are, in check right there, then you're not going to be sick. The problem is when you take a broad spectrum, let's say, again, we're going to stick with antibiotics. If you take a broad spectrum antibiotic, and that antibiotic targets your normal flora, killing your good, or harmless bacteria, but along the way, it can go ahead and target the bacteria that's causing you harm, okay? What happens is by killing a lot of your bacteria, normal bacteria, what ends up happening is these pathogens right here, they are going to now start replicating and taking over. So let me give you an example to make more sense of what I'm trying to say. Let's say you have someone who has a strep throat infection. Okay, so the infection is right there. They have a strep throat infection. And so strep throat is, what's a type of bacterium that causes a strep throat? Streptococcus pyogenes. So Streptococcus pyogenes, that is a coccus bacteria, and it's a gram-positive bacteria. Now, if someone has, a strep throat infection, what's a good antibiotic to give them? Well, we talked about it previously, and we said that penicillin, it's a good antibiotic that targets gram-positive coccus bacteria. And we said that penicillin is a narrow spectrum antibiotic, and it targets, gram-positive coccus bacteria. But let's say that your physician does not prescribe you penicillin, which is a narrow spectrum antibiotic specifically targeting gram-positive coccus bacteria, and one of those bacteria could be the one causing the strep infection. Let's say they don't prescribe a narrow spectrum antibiotic and they prescribe a broad spectrum antimicrobial. So they prescribe something that can kill what? Both gram-positive and gram-negative. Okay, what's the problem when a lot of broad-spectrum antibiotics are actually being prescribed instead of the narrow-spectrum ones? This is what can happen. When you're given a broad-spectrum antibiotic, it's going to kill way more bacteria than necessary. It can kill, of course, it's going to target the, in this case, it's going to target the strep throat infection. Okay, so some of these X's that you guys are seeing could be the bacteria, again, that is causing the strep throat infection. But at the same time, it's going to go ahead and start killing, your normal biota as well. And it's going to start destroying, a whole lot. And by the way, when you take an antibiotic, it's not just going to, so let's say this individual has a strep throat infection. Is it the antibiotic, is it only going to work like right here? No, it's going to start circulating, right? And I mean, plus you've ingested it, so already it's in your colon, and then it's going to start, circulating your system. And as you can see, it's going to go ahead and kill a whole lot of your normal microbiota. Now, yes, it's going to kill the strep throat infection along the way, of course, but as you guys can see, it's killing like a whole lot of unnecessary, normal microbiota that you have. Now, what is the problem still? Well, as you guys can see, the problem is some of these pathogenic bacteria that you have in your, in your system could be resistant, could be resistant to that broad spectrum antibiotic that you took. And so if they are resistant to that broad spectrum antibiotic that you just took, and now you are now at a disadvantage because you've killed a lot of your normal, a lot of your beneficial, normal microbiota, and now you're left with your pathogenic normal biota, because again, remember, yes, these are pathogenic ones, but they were still part of your normal microbiota. But now, because there aren't good bacteria that can keep them, kind of restricted and basically in check, what's going to happen is they're going to go ahead and spread. And now we have a problem, because now that pathogen, even though, yes, it is part of your normal microbiota, but now do you see how it takes over? And that's why it can cause, problems, and we refer to that as a superinfection. And these microbes right here, they will overgrow, and they will go ahead and start harming you. And so that's why you will notice that if you take an antibiotic, a lot of times when you read the kind of like that information page that's associated with your antibiotic, it will tell you taking this antibiotic can cause C. diff, Clostridium difficile infection. Because again, if you take this antibiotic, most likely it's a broad spectrum antibiotic and it can kill a whole lot of your beneficial microbiota along the way. And so the C. diff can actually start to go ahead and reproduce in high amounts within your colon, which is definitely going to be a major problem. If we take a look at some examples of superinfection, let's say that there's an individual who has a urinary tract infection caused by E. coli. Now, E. coli is a gram-negative bacteria. So what's a good narrow spectrum antibiotic that can target gram-negative bacteria? For example, polymyxin. So polymyxin is a narrow spectrum antibiotic that targets gram-negative bacteria, and they can definitely target, E. coli that causes urinary tract infections. So let's say that this individual, though, was not prescribed polymyxin, which is a narrow spectrum antibiotic that can target gram-negative, bacteria, and it can target, other bacteria that causes urinary tract infections that are gram-negative. Let's say they were not prescribed that one. Let's say they were prescribed cephalosporin. Cephalosporin is a broad spectrum antibiotic. So cephalosporin can target both gram-positive and gram-negative bacteria. So what happens is if this individual took a broad spectrum antibiotic instead of a narrow spectrum antibiotic such as polymyxin in this case, what's going to happen is the cephalosporin is going to target a whole lot of other bacteria, both gram-positive and gram-negative, not only E. coli. It's going to go ahead and kill off some other, bacteria that are part of your normal biota. And let's say that this individual who took cephalosporin is a female. And one thing to note about the female, vaginal region is there are other, bacteria such as lactobacilli, and lactobacilli is gram-positive. So when this individual takes cephalosporin, as I said, cephalosporin can target both gram-negative and gram-positive. So what's going to end up happening is those lactobacilli, they're going to get killed along the way. Now, these lactobacilli, when they go ahead and get killed, the Candida albicans, which is actually a yeast, okay, goes ahead and overgrows. The reason why Candida albicans was placed under check, sorry, the reason why Candida albicans was placed under check and not overgrowing because actually of these lactobacilli, because they were maintaining an acidic actually environment in the female vaginal region. So when those lactobacilli got killed, and now the pH in the female's vagina is going to change and it's not going to be acidic anymore, the Candida albicans is going to go ahead and start overgrowing. And what happens is when the Candida albicans goes ahead and overgrows, that's going to cause a superinfection. That's going to cause yeast infection, or it's going to cause an oral thrush. Another example, if we take a look at tetracycline, clindamycin, broad spectrum penicillin, remember how if you're going to say broad spectrum penicillin, then you're talking about those semi-synthetic, right, penicillins that they modified in the lab. So when you take such, broad spectrum antibiotics, as I said, you're going to kill off a lot of your normal biota. Okay, so when someone takes, again, broad spectrum antibiotics, what can happen is a lot of the normal biota in the colon can go ahead and start dying. Now, In the colon, there's also Clostridium difficile, or C. diff. But again, it's not causing any problems because a lot of the, harmless or beneficial bacteria that you have in your colon is basically putting this bacteria in check. It's not allowing it to overgrow. But what happens is if a lot of the, harmless or beneficial bacteria that are part of your normal biota dies, the C. diff is going to overgrow and it's going to start invading the intestinal lining, actually can start killing the intestinal lining by releasing toxins. And so the intestines can actually start dying. And that's pretty bad because if the intestines start to die, it's going to cause extreme abdominal pain, fever. The first symptoms usually, individuals get if they have a C. diff, infection is diarrhea. So they're going to get a lot of diarrhea and it's going to smell very, very bad. So, they would want to know, like, if you have diarrhea, like how bad does it smell? Because if you have a lot of diarrhea after taking a certain antibiotic and you tell them that it smells really bad, that's not a good indication at all. And then you later develop, abdominal, pain and then fever. And if the intestines die, the one problem is, of course, that's a huge problem in the first place. It's dead, and now it's going to have to be cut off. But the major problem is if this intestines dies and it goes ahead and bursts before they take it out. So again, it's going to have to come out because it's dead. But if it bursts before they take it out, then the individual gets a septic, goes into sepsis and they have a septic shock. And so that's why when you are taking those antibiotics, definitely recommended that while you're taking antibiotics, take something that has probiotics. It has like good bacteria, okay? So things like yogurt, things like milk, those have a lot of good beneficial bacteria. And what you're trying to do is, why should you be taking those probiotics while taking your antibiotics? To basically replace those microbes, those helpful microbes that you were killing along the way, because as I said, most likely you're taking a broad spectrum antibiotic that's killing like a whole lot of, unnecessary microbes that don't really need to be killed, but it gets killed along the way. So take probiotics to help you replace those microbes that are lost, along the way. And to kind of strengthen the biota that you already have. So things like, as I said, yogurt, milk are good options. Now, there are probiotic pills, okay, but if not, yogurt is a pretty good option. Now, when we think of these antimicrobial treatments, right, in vitro, meaning if you're trying that drug, let's say you're trying an antibiotic on an agar plate, okay, and you want to see how resistant or susceptible or intermediate your bacteria is to that drug. Now, you can actually get a totally different response on that agar plate compared to what you get in vivo, like when the individual ingests that antibiotic. And the reason why sometimes, that antibiotic works really well on an agar plate, but when the individual takes it, it's not going to work that well. So why is it failing when you go ahead and ingest it? Well, it could be so many different things. It could be that, this drug that you took it cannot diffuse properly, throughout your body. So it's not reaching the site of infection. Okay, so it's not going to work. Or if you do have resistant microbes and they didn't realize that you have those resistant microbes. So when they took a sample of the, like, let's say they took a urine sample from the patient and they cultured it, but they failed to see that the urine sample has some resistant microbes to, let's say, penicillin, and then they go ahead and prescribe this individual penicillin. Well, it's not going to work because they have resistant microbes that were not accounted for. Let's say that they have more than one pathogen. Okay, so if they have more than one pathogen and you prescribe, let's say, a specific antibiotic, and that antibiotic doesn't target, that other pathogen group, then again, then the antibiotic is not really going to work that well. Or maybe the individual is not even taking the antimicrobials. And again, antimicrobials could be anything. It doesn't always have to be an antibiotic, but that's what we are mostly focused on. So maybe they're not even taking it correctly. Okay, maybe they're skipping dosages. Maybe, again, they didn't finish the course of the antibiotics. Maybe maybe they're taking some other drugs that contradict these antibiotics, and so therefore the antibiotic is not functioning properly. So there could be, again, various reasons, when it comes to how the patient is not taking it correctly. Now, they also found out that bacteria that survive, in biofilms. So as we said, previously, in previous weeks, we talked about biofilms, and we said that bacteria can survive on, on surfaces, and they create this community, right? And they, they even travel as a community. If they're going to, travel, then the whole community kind of takes off together. Now they found out that actually bacteria living in a biofilm community is going to act differently compared to if that bacteria is living on its own. So they actually found out that bacteria, when they were trying to target a certain bacteria in a biofilm, let's say they were trying to kill a certain bacteria in a biofilm using an antibiotic, they actually found out that they have a hard time killing a bacteria that's found in a biofilm compared to killing it if it's on its own. If you think about it, it's like, why is it hard to kill it if it's in a biofilm? Because it has its community, right? It has those other microbes surrounding it that actually help it out. And they found out that, when those bacteria are basically together, they can even express different phenotypes and different genetic characteristics about them. And so therefore they found out that it's way harder to kind of kill bacteria in biofilms compared to if that bacteria was on its own. Now, if we talk about agents that do treat fungal infections, as we said, fungi are eukaryotes. So those drugs that we mentioned, like polymyxin, cephalosporin, carbapenems, etc. Now, though, can I take those to go ahead and treat a fungal infection? Absolutely not. They're not going to be effective because, again, fungi are different than bacteria. Now, there are similarities, like they both have, if we talk about similarities between fungi and bacteria first, I mean, they do have a cell wall, they have a plasma membrane, but structurally it's totally different, right? So structurally, the cell wall of a fungus is totally different than a cell wall of a bacteria. So even though, an antifungal can still target the cell wall, that antifungal is targeting specifically the cell wall of a fungus. Compared to if you take an antibiotic that targets the cell wall, again, it's going to be specifically targeting the cell wall of a bacteria. So that's why antibiotics will not work if you have a fungal infection, because antibiotics are specifically against bacteria. And fungi, though, they're eukaryotes. And if you think about it, our human cells are eukaryotes. So there are similarities between between fungal cells and human cells. And that's why that kind of makes it then hard, right? Because a lot of these drugs then could, that are toxic to the fungus can actually harm our tissues. Because if you think about it, we're both eukaryotes. So there are going to be similarities. And so some of these drugs can actually harm the human tissues. And that's why fungal infections are pretty hard to get rid of. And this is kind of one reason why. So fungal infections, again, they're pretty hard to get rid of. So far there are only like a few agents that we have antifungal properties. So in terms of how many antifungal drugs do we have out there, it's not a whole lot. Okay. If we talk about agents that, antimicrobials that basically treat viral infections, I mean, viruses are their own group, right? So some of these agents, the way they work is they can prevent the virus from entering into your cells, okay? Some of them, they go ahead and they prevent the virus from using your enzymes to go ahead and replicate and make more of its own, DNA, etc. So this is how it works against it. Some antivirals drugs, they go ahead and they prevent the virus from assembling itself. So it prevents the virus from putting itself together, okay? And so again, this is how it affects it. Or it prevents the virus from getting released out of the host cell in the first place. So these are, some things that could be targeted if you want to go ahead and, kill the virus. This is how kind of like antivirals work. Now, we also talk about drug resistance. So we said that drug resistance, this is when the bacteria becomes, it's tolerating that drug that you're taking, and it's, tolerating it, meaning it's not being really, killed by it, and it can, so meaning it's kind of resistant to it, okay? Now, why is that problematic? Because that resistance could actually spread to other bacteria. And by the way, that resistance could be intrinsic, meaning that the bacteria itself has a gene that makes it resistant to that antibiotic, let's say, that you are taking. Or it could be acquired, that you have a bacteria that didn't have resistance to a certain antibiotic, but it gained it. It gained it from another bacteria. So that resistance, again, could be acquired, or it could be, intrinsic. It could already be, the bacteria is already carrying that resistance gene. So let's take a look at, this image right here. So if you remember, we said that bacteria, they can have their own, DNA. So let's say here is the DNA. But then bacteria could also have these extra pieces of DNA called plasmids. Now, these plasmids, they are basically, DNA information, and within that plasmid, the bacteria could carry a bunch of resistance genes. One of these resistance, kind of genes that a bacteria can carry, we refer to it as the resistance factor, the R factor. So let's say that this bacteria right here, let me just choose a different color. So let's say that this bacteria right here carries, oh, you can't really see it. So let's go with yellow right here. So let's say that this plasmid right here, okay, carries that R factor. Okay, so it's basically carrying that R factor, which is basically that resistance, factor. So right here, R. Okay. And let's say that this R factor right here, it is carrying an antibiotic resistance gene. Now, depending on what that antibiotic resistance gene, could it be a gene that's resistant to only one antibiotic? Yes. Could it be a resistance gene that makes this bacteria resistant to multiple antibiotics? Yes. Okay, so let's say that this bacteria right here carries a resistance gene against penicillin. So if you try to kill this bacteria using penicillin, it's not going to work because this bacteria is resistant to penicillin. Let's say that this bacteria right here, okay, doesn't have the plasmid, okay? So if this bacteria doesn't have the plasmid, that means this bacteria is not resistant to penicillin. So what happens is this bacteria right here can actually transfer over its plasmid. It's not going to completely give it, but it's going to like give it part of it, okay? So via the conjugation, okay? So remember these conjugation pili that we again talked about in previous weeks. So this bacteria sends off its, conjugation pili, and it can go ahead and let me draw it for you. It can go ahead and transfer over. Okay, it's actually going to like copy its plasmid and then give it to it. So so now they're both going to have what? Now they both have that resistance, now they both have that resistance factor right here. Okay. So now, instead of having only one bacteria that's resistant to, let's say, penicillin, now you have two bacteria that are resistant to penicillin. And do you see how this problem, again, can go ahead and spread? So do you see how now it's going to go ahead and actually, via its conjugation pili, spread it to this bacteria as well right here? And so, and you guys see how the problem, starts getting out of hand. And so therefore, this is how then we end up with a lot of bacteria that are what? A lot of bacteria that are antibiotic resistant. So as you can see, this bacteria right here already had that intrinsic, right, resistance. It had it within its plasmid. See how this bacteria basically acquired it, okay, via the conjugation pili. Or, what's another way for bacteria to acquire antibiotic resistance? Via transduction. Remember when in previous weeks when we were talking about bacteriophages, and we said that these are basically viruses that can attack bacteria, and we said that those viruses could be carrying information from an old bacteria host. And then as they infect the bacteria, they go ahead and they inject that new, new characteristics. If you guys want, you can go back to that virus, video and kind of like, refresh your memory about it. But we talked about how transduction is also another way for bacteria to end up acquiring new characteristics, okay? And so this is why we are facing a huge problem and why we are having a lot of antibiotic resistant bacteria out there. Because let's say that here you have a bunch of bacteria and a few of them are resistant to that antibiotic that you just took. And, when you took an antibiotic, it was able to kill, a lot of bacteria, but I mean, it wasn't able to kill those resistant ones. So what ends up happening is, these bacteria that are resistant to the drug, they go ahead and they share their resistance with everyone else, right? And so instead of having just two that are resistant to that antibiotic, now you have a bunch of them. And the problem, grows and gets out of hand. And the CDC is kind of warning that, the problem is really getting out of hand, and we now have a lot of these superbugs. And, they call them superbugs because they are resistant not just to one antibiotic, but they are resistant to almost like all the antibiotics that we have. Why? Because if you guys have, let's say, let me go ahead, kind of erase this part, and let's say you guys like this bacteria over here, okay? Let me just grab a different color. Okay, so let's say, guys, like this bacteria over here, okay, also has a plasmid. And let's go with a different color. Okay, so here, purple. Okay, so let's say this bacteria right here has this resistant, gene, but let's say it's resistant against a different type of antibiotic. Can it go ahead and actually pass it on to, pass it on to that other bacteria? Yeah, so here we go. So it can go ahead and pass it on, to this bacteria right here. And what ends up happening is now this bacteria is not just going to be resistant against, only one, antibiotic. No, guess what? It's also going to now be resistant to that other antibiotic right there. So you can end up then having one bacteria that's now resistant to a whole lot of antibiotics. Okay, now we talked about how, we're going to have bacteria that are resistant to some antibiotics that we have out there. Now, if you think of the mechanism, okay, so what's really happening? Why are they resistant to this antibiotic? Well, they could be carrying enzymes that could make those antibiotics ineffective. So, for example, if you have a bacteria and you're trying to kill this bacteria using the penicillin antibiotic. But this bacteria that you're trying to kill using the penicillin antibiotic has this enzyme, penicillinase. What this enzyme does is it breaks down the beta-lactam ring of penicillin, making penicillin ineffective. So this is one way. What's another way? Well, the bacteria goes ahead and mutates those receptors that it has on its surface. And again, this is not only happening in bacteria. I mean, it could happen, with other organisms, microorganisms as well. But again, we're focused on bacteria. So what could happen is, the bacteria could go ahead and mutate those receptors that it has. And so these receptors are needed for the drug to go ahead and attach to it, so that the bacteria can die. Well, if they mutate those receptors, the drug is not going to be able to bind to that receptor, and so therefore the drug cannot enter, and therefore the bacteria is unharmed. Another way is the bacteria goes ahead and kind of, again, through mutations, it's going to have this kind of pump. So when the drug gets into the cell, guess what? It just pushes it back out. Okay, so drug comes in, pushes it really quickly out. Another way is, we talked about how some drugs out there can go ahead and target the ribosomes, right? And basically to kind of prevent the protein synthesis. We gave examples like azithromycin. We also talked about tetracycline, right? And those are, antibiotics that target the ribosomes. Now, what ends up happening is the bacteria basically can go ahead, again, through mutations, it can go ahead and alter, alter its own ribosome. And so therefore, if it alters its own ribosome, guess what? The drug cannot now bind to the ribosome. See right here how the drug is attaching to that large subunit. But notice how now the ribosome, has a different shape to it. So guess what? The drug cannot bind. So these are some, mechanisms that take place that will, contribute to why those bacteria are resistant to certain antibiotics.